All right. Hello, everyone. I am feeling a little bit better, so let's jump into this exam 5 practice. So here now we're gonna jump into the practice exam and go over the answers to that. So yeah, let's just hop right into the true or false. So okay, I'm having a bit of a technical difficulties here. If you'll give me 1 second. Okay, let's make this a little larger. Yeah. I'm not sure why it changed on me. Hmm. I guess we'll just have to do. Alright, so, number one, bacterial chromosomes are typically double stranded circular DNA molecules. So, this is going to be true. So, why is this true? So, basically, bacteria do not have nuclei. They do not have a nucleus in their cell because they are prokaryotes, right? Only eukaryotes have a nucleus. So, prokaryotes also only have just one chromosome. I don't know if you could really call it a chromosome. They just have one set of like DNA, and it's in the form of one giant circle, right? Now, whether or not this like circle like goes around like this and then finishes here, well, this would still be considered one DNA strand, right? Or if it actually just goes around like this, which is less likely. No matter what, these chromosomes do not have. Sorry, these sorry, it says bacterial chromosomes. Yeah. So these chromosomes do not look like they would in eukaryotes, right? Where we have a lot of distinct chromosomes in the sort of X conformation. So bacteria are going to have more like a circle, right? So bacteria do not have nuclei, but they do have nucleoids, right? So nucleoid, because they don't have the membrane of the nucleus, but the DNA has to kind of sit in the same region. They call it the nucleoid because it mimics a nucleus. So outside of this region, so let's say let's say this pink region, let's say this is our nucleoid, right? And then outside of our nucleoid, we have what are known as plasmids, right? So these plasmids can go out. So let's say we have like this cell, right? Let's give it like a little flagellum too. So these plasmids can actually go out of the cell and influence their environment. So they could pump these plasmids into other bacteria or into eukaryotic cells. They could pump these. They could essentially not necessarily infect, but they could give other organisms these plasmids, whether or not it's beneficial or not, but they can. So that's the kind of advantage of having these plasmids versus having regular chromosomes. So we have the nucleoid region, and then we have these plasmids. So moving on. Um, all right, number two. Okay, I've had a feeling that was going to happen. All right. So for whatever reason, yeah, for whatever reason, Microsoft Paint does not want to let me do this. So here's what we're going to do. Bam. All right, just like that. So number two is asking, chromosome is the structure adopted by chromatin during metaphase. So this is going to be, sorry, this is going to be true. So why is this going to be true? So there's a lot of vocabulary you have to know, right? Chromatin, chromosomes, DNA, nucleotides, all that sort of thing. They're all related to each other, right? So for now, let's go over chromatin versus chromosomes. So basically, chromatin. I don't know why the font's like this. It looks so weird, doesn't it? Okay, let's just go for good old Times New Roman. Yep. Okay. So, and let's make it a little bigger too. So chromatin is made up of DNA plus its associated proteins. A chromosome is made up of chromatin. All right, so what does this mean, right? So a chromosome is basically just like a packed up molecule of DNA plus proteins that contains genetic information, right? So it contains the genetic information, and in humans we have 46 chromosomes, half of which come from mom and the other half come from dad, right? So that's a chromosome. How about chromatin, right? So chromatin makes up chromosomes. So yeah, when we say chromosome is the structure adopted by chromatin during metaphase, well, chromatin makes up chromosomes, right? So DNA, right? That's of course like A, T, C, and G, right? And its associated proteins. So these associated proteins would be something like histones or like promoter regions, or maybe not promoter regions, more like activator proteins, that sort of thing, right? Like it's just there's a lot of proteins that exist within the DNA to aid with transcription or DNA replication, that sort of thing. So that's all the associated proteins, right? So that's chromatin, right? So the answer to this one will be true. So moving on. So we just kind of went over this, right? This is kind of redundant. So number 3 is asking chromatin is made up of DNA and protein. And of course, that is going to be true. So the DNA, of course, I mean, that should be obvious, right? A, T, C's, and G's, right? That's the DNA that we've been familiar with for some time, that we have been familiar with for some time now. And the proteins, which we just went over, which were like the histones and other proteins that exist on the DNA. So that's number 3. So let's move on to number 4. My computer's really been acting up lately, so let's see. So number 4 is asking, histones are rich in aspartic acid and glutamic acid. All right, this one is going to be false. Now, why is it going to be false? So the thing about histones is that histones are negatively charged. So now I ask you this, right? Um, sorry, I totally messed it up. Histones are going to be positively charged. Ignore that previous statement. Histones are going to be positively charged. So what charges your DNA, right? So DNA is of course going to be negative. So we could look at that in a second. Um, so let's look here. Yeah, this should be good. So here we go. Here we have your 20 amino acids. Well, this is 21 because this has the addition of selenomethionine. Sorry, selenocysteine. Sorry. In any case, let's look at our amino acids. So here we have the positively charged side groups and the negatively charged side groups. So these will look kind of confusing, actually. I think I'm going to try and find a different one. Okay, yeah, this is much better. So here we have 4 different groups of amino acids, right? And these is what make up your proteins, right? So histones are proteins. So we have the nonpolar, the positively charged, the negatively charged, and then the polar. So negatively charged, okay, well, what's that going to be? Aspartic acid and glutamic acid, right? So whenever you see aspartic acid, if you ever see some sort of acid, it's always going to be negatively charged in our bodies, right? Or if it ends with ate, like in the case of aspartate or glutamate, it means it's going to be negatively charged. So remember, histones are what bind together protein. That's what protein. Sorry, histones are what bind together DNA. DNA wraps around the histones and it keeps it all together. It keeps it all condensed into one spot, I guess. So DNA, remember. So if we draw just like a basic nucleotide, right? And then let's do that. Yeah, not super great, but it'll have to do. So this is like a basic nucleotide, right? So now this is going to be a phosphate group, right? So what does the phosphate group look like? So by itself, the phosphate group looks like this, right? So these are all negative charges. So that's like no good, right? Or sorry, oh my gosh, I'm. It's been a bit of a long day. Um, I keep on messing up just minor things, so you'll have to bear with me. So it's not necessarily good or bad that these are negatively charged. It's just how it is. So if we ever see like this phosphate, if we ever see like the P with a circle in it, that means phosphate group. So we have the phosphate group here, and those are going to be negatively charged. So opposites attract, of course. So that's going to be attracted to like a positively charged. So histones, right? Histones are going to be rich in amino acids that have positive charge, like lysine, arginine, or histidine, as we see here, right? What they're not going to be rich in is aspartic acid and glutamic acid because those are going to be negative. Those are going to be negative. So the answer number 4 will be false, right? So for that one, you have to understand what histones are. So I hope we know that. If not, so yeah, basically histones are just like what keep together DNA. Which I just wonder for that. Sorry, like I said, it's been a bit of a long day. So in any case, so okay, well this one just tells you the answer. So the large ribosomal subunit is made of rRNAs, while the small ribosomal subunit is made of proteins. And of course, it already says. So this is going to be false, right? So why is it going to be false? So basically, ribosomal subunits, what those are. So let's do it in brown. I always think of ribosomes as brown. I don't know why. Is basically we have like two of these subunits, right? And let's fill it in too. Why not? Um, and let's make our RNA. Let's make our mRNA blue, right? So this is our ribosome, right? So the ribosome has two portions to it, two subunits. So what that means is that that means when RNA is not bound to ribosome, these are going to be kind of separated like this, right? So they're going to be separated. They're going to do their own thing. But when they're together, then it makes one ribosome, right? Two subunits for one ribosome. So, what are ribosomes made out of? Well, ribosomes are primarily made out of rRNA. So now rRNA is unlike rRNA is not like tRNAs and it's not like mRNAs. They're kind of completely different. The only purpose for rRNA is to make up a ribosome. And This goes for both the top portion and the bottom portion. So it's not like one is made of one is made of rRNA, the other one is made of protein. No, they're both made of rRNA. However, they also both have proteins. So that's going to be the top portion and the bottom portion. Once again, it's not like an either or thing. It's just like they're both made of rRNA and they're both made of protein, and that's going to be your ribosome. So rRNA doesn't have anything to do with like transcription or translation. It just has to do with what the ribosome is made out of. Right, so yeah, moving on. So that one's false, right? Moving on. Let's see, number 6. So if you may notice a trend, we have the first 3 that were true, and then these last 3 were like kind of false, right? So I'm going to try and speed through this a little faster because I feel like I'm taking forever. In any case, so this one's going to be false. The question is asking 2 ribosomal subunits joined together before the ribosome binds to the mRNA. So let's draw our ribosome again. So for our ribosome, we have the bottom portion, the top portion, and the mRNA. Right, so basically, we have the bottom portion. Let's say B for bottom, right? We have the bottom portion. The mRNA comes in, and it binds here, right? And then once it's bound here, with the top ribosomal subunit comes in and sees this and says, "Hey, I want to get on that action." So it comes in and plops on top of there, and then we can start translation. So then we have the tRNAs, right? So then we have the tRNAs. With the amino acid, right? And they come in, and they go out, right? And they're making a polypeptide chain. That's translation, right? So what I'm trying to get at is we have the bottom portion first. We have the bottom portion first, the mRNA comes in, and then the top portion second. Or the top portion last, I guess. So it's not like the ribosome is something that's joined together before the RNA binds. We have the RNA binding before the ribosome joins together. So that's what's going on there. So the answer is false for that one. So the next question is asking number 7, right? Once again, actually, never mind. So this one was one of your Slido questions on Monday. Number 7 is asking, neutral mutations are typically selected out of populations since they do not alter phenotypes. So this one is going to be false as well, right? So what are neutral mutations? Neutral mutations, right? It essentially just means that there is no change. There's most likely no change in a phenotype. So let's one second. So let's use our amino acid chart as an example, right? Let's say we have an amino acid that codes GCU, right? So that codes for alanine, right? But then we have an amino acid that codes for GCA. Well, guess what? That also codes for alanine, right? So basically, this redundancy is typically used to catch some of these neutral mutations because so any sort of mutation, any sort of mutation kind of just happens spontaneously, right? Whether it be good or bad, or in this case neutral, right? So basically, there's nothing changing about the actual phenotype. We have still the same amino acid, right? So nothing's actually being changed. So why is this important? Or why is this important to the question? So basically, if there is a mutation that is detrimental to the organism, then it's typically going to be selected out of a population. Or if we have a mutation that is aiding in the survival of an organism, it is going to typically stay within a population due to survival of the fittest and whatnot, right? That's just basic Darwinian evolution. So what happens therefore if there's neither good nor bad coming out of a mutation? Well, this question is saying that they will typically be selected out of populations. This is true. This last portion is true. They do not alter phenotypes. That is true. But because they do not alter phenotypes, they will not be selected out, right? Because there's nothing necessarily keeping it in or out, right? If a trait, if there's a neutral mutation, then there's a trait that has been changed that is not necessarily good or bad for the individual. So therefore, it's not going to be selected in or selected out. It's just going to stay or it's going to go. It essentially just happens randomly. There's no necessarily increased chance of it staying in or out. So that's number 7. It's false. So number 8. Okay. So we're gonna do number 9 and number 8 together because they're kind of related to each other. Okay, so lac I is located in a regulatory region of the lac operon. Number 9 is saying a group of co-regulated operons is called an operator. So, if you'll give me one moment. Okay. So this is our lac operon, right? So I don't like this question because it feels like more of like a genetics question than it does a biology than it does like a molecular bio question. Um, I would get questions like this when I was in genetics, and guess what? Dvork was also teaching that class. So I apologize, but I have a hard time with this as well. So you'll have to bear with me. So number eight's asking the lac I, or I think that's lac. Yeah, the lac I is located in the regulatory region. Okay, well let's look at our lac operon. Okay, so yeah, here we have a color coded diagram here. We have the in the light blue we have the lac I, lac Z, lac Y, and lac A, and all of these regions are coding for proteins. So the lac I is therefore not going to be in the regulatory region because look, in this green we have the regulatory regions P, I, O, and P. So these regions are actually what's going to be regulated. These regions code for proteins. So whenever we see lac I, lac Z, lac A, lac Y, these are regions coding for proteins, not regions that count for regulation. So number 8 is going to be false, therefore. Okay, so then number 9 is asking a group of co-regulated operons is called an operator. Well, that's not true because a group of co-regulated operons, I believe, is called a regulon. So I don't know about you guys, but I don't remember learning that about that either. So I really apologize that this is just reality for you guys because like some of these questions are just kind of ridiculous, honestly. Once again, I really don't like this question. Number 8 and number 9 kind of suck because I really hate just the lac operon and I hate like the little specifics of it. Co-regulated operons called operator, that's just false. So no difference between operon and operator and then operon and regulon. So yeah, we can go over that a little bit now. So hmm, So for example, like this would be like, oh, actually, never mind, ignore that, ignore me. So I'm just gonna take this from Google. If you search up Regulon versus Operon, so in prokaryotes, an operon is a functioning DNA unit composed of several genes that are controlled by a single promoter and operator. On the other hand, a regulon is a functional genomic unit comprising of a set of genes that are not necessarily contiguous and are regulated by a singular regulatory protein. So yeah, if we have a group of co-regulated operons, right? So if they're all co-regulated and they're all regulated by one protein, right? And that means they're not necessarily contiguous. So that's consistent with the definition of a regulon. So this would not be an operator because an operator kind of is part of an operon. So this would not be considered an this would not be considered an operator. This would be considered a regulon. So yeah, like regions like this would be so this is the lac operon, right? So this is controlled by like this this protein, right? This regulatory protein. Um. Anyway, I'm really sorry, guys. This is not going very well. You have to forgive me. This is not my favorite unit in the world. I don't know if you can tell. Hopefully, or at the very least, it'll be over tomorrow, so we can jump into something else on Friday. In any case, so number 10 is asking microRNAs increase the expression of mature mRNAs. So this one's going to be false. And now why is it going to be false? So microRNAs are a pretty recent discovery in terms of like actual genetics and gene expression. Um, there were like I said, pretty recent discovery. Um, so basically, what microRNAs do is that they act as like sort of checkpoints. They see, so if we have like an mRNA, right? We have like microRNAs that come in and see, all right, if everything's working just fine. So basically, they act like checkpoints. So if they say everything is okay, everything's good, right? Then there's no problem, right? Or if we have one that says, okay, this one's bad, and this one's bad, and this one's bad, and this one's bad, and this one's good, why not? If we have something like this, then then it's going to actually hold back the mRNA from further translation. It's going to see these errors in the DNA, in the mRNA. It's going to see these errors and it's going to try and regulate those errors, try to either remove them or delete the mRNA. So this is like the opposite of increasing the expression. This is like a sort of like border guard or security checkpoint. It's actually increasing traffic, if anything. It's not increasing the expression. So this answer would be false, therefore. So moving on. So like I said, microRNAs are pretty recent discovery. They we didn't yeah, I don't know where I'm getting with that. It's because they're so tiny, right? It's kind of hard to yeah. Okay, I really wish like the text prompt would actually work properly. It's really kind of frustrating me. Okay. Okay. So number 11 is asking genes are transcribed into complementary A non-overlapping nucleotide codons of mRNA. B overlapping two nucleotide codons in a stop nucleotide. C non-overlapping three nucleotide anticodons of tRNA, or ribosomal RNA. So right off the bat, we can go ahead and eliminate D, right? Because ribosomal RNA is only we only care about that within actual ribosomes, right? So genes are transcribed into complementary non-overlapping three nucleotide anticodons. So we're not transcribing into tRNAs, right? We're using tRNAs to translate, right? So tRNAs, we don't even want to worry about this, so this one's going to be wrong too. And overlapping two nucleotide codons and a stop the nucleotide, well, that one's wrong because we know that mRNAs operate in threes, right, not twos. So that leaves only A, right? Non-overlapping three nucleotide codons of mRNA. So, moving on. Okay, well, like, what is a codon, right? So, yeah. So let's say we have our mRNA, right? So this is our mRNA. So let's say we have AUG, CCG, UAG, AAA, and etc., right? So codons exist on the actual mRNA, right? They exist on the actual mRNA, right? They don't they don't necessarily exist on the template strand because we're not really worried about template strands. We're not really worried about codons in the template strand, right? Because like when we're transcribing mRNA, it just kind of does it indiscriminately, right? But once we actually have the final product, then we could subdivide into different codons. So codons, right, we have these three nucleotide regions that each code for it once we go to translation. Once we go into translation, then will actually give us a specific amino acid sequence, right? So each one of these three codes for just one of these. So AUG, that's the most popular example, codes for methionine. CCG, I'm not sure for the rest of these. I just use this as an example. So basically, that's just a codon. It exists on the mRNA. It doesn't exist anywhere else. It just exists on the mRNA. That's what a codon is. And then, so like the number, like letter C, answer C is asking non-overlapping three nucleotide anticodons of tRNA. Well, what is an anticodon? So basically, right? So when we have our tRNAs, when we're going into translation, right, we have the tRNAs, and then so the tRNAs have to bind to the codons, right? How do we do that? Via an anticodon. So if we have the mRNA, so if we start at AUG, right, we have to have an anticodon that says the opposite. So it would have to say UAC on the anticodon. And essentially what that just looks like is if we have our tRNA, right? And then we have the amino acid on one end. And then we have this anticodon on the other. So think of it like a USB port. I don't know why it reminds me of that, but think of it like a USB port. So obviously we're gonna have like the female end of the USB port, and then we're gonna have the actual USB. We can think of this as the USB, right? Because we're fitting it in, and like how it's like poking out like this, right? So basically we're fitting it in here. So that's what an anticodon is. It just says the opposite of what the codon does. But it's also important to note that we always read that when we have like the codon chart, we always read the codon, not the anticodon. Unless you have an anticodon chart, in which you would read the anticodon. Anyway, we don't have to worry about that because we're only going to get a codon chart, right? It's also important to know that you have to know, excuse me, that you know the difference in what you're looking at. So right now we're looking at mRNA, but what if we had, for example, the template strand? Then you'd have to know how to transcribe from the template strand, and then translate from the mRNA. that sort of thing. So yeah, this is so when we're talking about tRNAs, this is what we're talking about, and when we're talking about codons, this is what we're talking about. So moving on. So there was a little bit of a confusion with this question, um, both for me and for you, right? This is what it says on the answer sheet. So the answer is not B, right? And on your sheets it shouldn't say B either, right? So the answer is not B. So that's what it indicates here, right? If you'll excuse me for one moment, I have to really blow my nose. I'll be back in 1 minute. All right, I'm back. Where was I? Okay, so basically I was just saying B is gonna be false, right? So let's first off let's read the question. So this one, so B is bolded, but just know that's not the right answer. The answer is actually gonna be D. But let's forget we just said that. So number 12 is asking the stop codon indicates A, the end of the promoter region and the beginning of the coding region. B, the end of the transcriptional unit. C, codons that form a ribosome. Or none of the above. So let's start from C. Codons that form a ribosome. Well, that one's just wrong because codons don't form ribosomes. That's rRNA, and we don't care about rRNA because that's not something that we really worry about in terms of transcribing. So B is saying that the stop codon indicates the end of the transcriptional unit. So this answer would be correct if it said the end of the translational unit, right? So this was actually initially a typo, right? But it actually ended up working out because of the answer option D, right? So stop codon indicates the end of the transcriptional unit. That's false because stop codon indicates end of translational, right? So then A is also saying the end of the promoter region and the beginning of the coding region. That's not right. Um, that's just wrong because this would be during transcription, not translation. So stop codon, like we said, we only worry about that during translation, so the answer would therefore be D, right? Not B. So yeah, basically, so we have the stop codon. So okay, so basically during transcription, we have promoter regions and terminator regions. Then during translation, we have start codons, or start codon singular, and stop codons. So basically, transcription and translation kind of have a bit in common because they happen in 3 steps. So transcription translation happen in 3 steps. So we have the initiation sequence, then we have the elongation sequence, and then we have the termination sequence. Right? So So because basically, right, we have to initiate transcription, and then we have to elongate the mRNA, and then we have to terminate the sequence so we don't go over what we need. That's the formation of the mRNA, right? And for translation, we have to initiate the start of translation. We have to elongate the amino acid chain, and then we have to terminate the amino acid chain once we hit the stop codon. So these two processes happen differently, but they happen in the same three steps. So what I want to emphasize with this is that during transcription, we have promoter regions, right? So promoter regions would be for initiation, right? And then terminator regions would be for, of course, termination, right? That makes sense. And then during translation, we have the start codons, which would be the initiation step, and then stop codons, which would be the termination step. An elongation in the middle, we have, RNA polymerase. And then we have that's for transcription. And then we have just regular codons for translation. Right. So that's just some like the processes that you have to or that you should know. Just know that the stop codon indicates the end of translation, not transcription. So the answer will be D. So currently we're at 41 minutes, about almost 42. Like I said, I just kind of want to hurry this up a little bit because sometimes I feel like these take forever, and you guys don't want to sit through all of this, I'm sure, because it can be quite exhausting. It is, trust me, it's exhausting for me too. Um, in any case, we can continue to go over these. I mean, of course, I don't mind it because I love to help you guys, but at the same time, it's like, damn, sometimes these take forever. So, for this question, I'm going to be honest with you guys. I don't really know what a ribozyme is. So, I'm just going to read it from Google verbatim, right? And I know, right? Like, so, this is the definition from Google. So, ribozymes are catalytically active RNA molecules or RNA protein complexes in which solely the RNA provides catalytic activity. The term ribozyme refers to the enzymatic activity of the ribonucleic acid nature, and it refers to the enzymatic activity and the ribonucleic acid nature at the same time. So ribosomes are okay, yeah. So basically, what this is saying, I think, is just to differentiate between ribozymes and just regular enzymes. So ribozymes happen they are ribosomes for one. They are in ribosomes. They have rRNA and then protein complexes within the RNA. I don't know how much you guys learned about that within the slides. If I'm gonna be real with you guys, I don't remember it ever being mentioned. I'm sure it was mentioned once or twice. However, that's not really the answer for this question because the question is asking the match between a tRNA and an amino acid is done by, and I know only some of you maybe are going to be biochem majors, so um, the pronunciation of this enzyme is a little bit difficult. Um, However, repeat after me: aminoacyl tRNA synthetase. Now say it again: aminoacyl tRNA synthetase. Synthetase. So yeah, even I mess it up, right? So basically, what is this? So What is this word saying? Whenever we say aminoacyl tRNA, you could just shorten that in your head to just regular old tRNA. So what does a tRNA look like, right? We've gone over this a couple of times now. See, I don't know why I have so much trouble drawing it. And then we have the anticodon. So this is our tRNA, right? So Oh wait, actually, this is wrong. So this is our regular old tRNA, right? So notice how it doesn't have an amino acid. So when we say the match between a tRNA and an amino acid, what we need to do is we need to actually attach the amino acid, right? So that's our amino acid. We need that to come in here and attach here. So now we have an active tRNA. So what does this? Well, it's probably going to be the thing that says amino acyl tRNA synthetase. So we're putting the amino group. So this is our amino acid, right? We're putting this amino acid on the acyl position of the tRNA, and we're using that via synthetase. Now, what the hell does that mean? Basically, what it just means is that we're attaching amino acids via an enzyme, and that enzyme is known as a synthetase. Not a ribosome, because that's where we attach the tRNAs to the mRNA. However, we're talking about a tRNA to an amino acid to a tRNA, and it's not going to be ribozymes either. So it's going to be the amino acyl tRNA synthetase. So number 14 is asking, a lac operon is irrepressible operon, an inducible operon, all the above, none of the above. Okay, so for this question, we need to know the difference between repressible and inducible. So let's go over that. So inducible versus repressible. So just by the language, what does it mean to induce something, and what does it mean to repress something? So to induce something means to like initiate something, to like start something, or to like put something onto someone. Induce, right? And then repress means to stop something, to prevent something from happening. So just from this context, right, we can indicate that inducible operons mean that we have to add something to get it to work. Versus repressible operons, which mean we have to add something to get something, add something to get it to stop working. Slash turn off. And for this one, I'm going to actually have a little addendum here, slash turn on. So we could also say that inducible operons are always off, or rather normally off. And then repressible operons are normally on. So that's kind of like a lot of words, right? Or I guess we could add one more thing, right? So this is going to be the lag operon. And this one is going to be the trip operon. So what do all of these mean? So operons, for one, only exist in prokaryotes, right? Gene regulation Gene regulation in eukaryotes is a little more complex, believe it or not. It involves a lot of proteins for one. Not to say that this doesn't already involve a lot of protein. Anyway, so when we're saying an operon is inducible, we're basically just saying like we said here, for the inducible operon, we have to add something to get it to turn on, and so that means it's normally off, right? So for in the case of the lac operon, well, lactose is not a readily available energy source, right? So if you're constantly making the enzymes to break down lactose when there is no lactose, right? Then you're just wasting resources. So that's the case of the prokaryote, right? If we don't have lactose as an energy source, then we don't need to break it down, so we don't need to have the enzymes for it. But when we do have lactose, what lactose does, lactose is a cofactor. What lactose does is it binds to the inducible operon, gets it to start working and turn on, and then start making those enzymes that break down lactose, right? So that's what the inducible operon is doing. That's what lac operon is. Versus repressible, so the repressible operon will be tryptophan. That's one example, right? So we always say lac and trip operons for these two examples because they're the most clear examples. That being said, the trip operon, so trip is short for tryptophan, right? Tryptophan is an amino acid that is pretty much always needed within prokaryotes and in case of most proteins, as a matter of fact. So we need tryptophan to be made. However, once we've made enough tryptophan, right, tryptophan acts as a cofactor for the tryptophan operon, right? So once we actually add tryptophan, tryptophan acts to then therefore repress the genes that make tryptophan, right? So I'm not really sure if it's like a negative feedback loop, but essentially we make this stuff and then once we've made enough, we stop making it. That's basically what it's saying. So that therefore we could say it's normally on. So then we have to add something like tryptophan to get it to stop working or to turn it off. So with all of this in mind, we can eliminate the lac operon being repressible. We could read letter C as an option. Letter C is saying lac operon is an inducible operon since the repressor will detach from the operator in the presence of lactose. That is correct. So the answer is C. So, moving on. Ah, damn. Please bear with me for one moment. Okay. Okay, so number 15. Let's check our time real quick. Currently we're running at 53 minutes, about halfway through. So yeah. So number 15. For this one, you have to know what a histone is, what an acetyl group does, what an acetyl group is, and what an acetyltransferase and deacetylase means. So the question is asking, which of the following might occur if an activator protein is bound to an enhancer related to chromatin structure? So the question is asking about binding an activator protein to an enhancer. Sorry, this is a very difficult question if you don't really know what's going on. So what is an activator protein, right? So an activator protein is the protein that starts transcription, right? So if we're binding the protein to an enhancer related to the chromatin structure, So if we're worrying about the chromatin, then we have to worry about the proteins and the DNA, right? Proteins being histones, right? So if we are enhancing transcription, therefore we have to do something that will loosen up the chromatin. So remember, histones, so DNA is negative, right? Histones are positive, and acetyl groups are negative. So, using this principle, right, if we need something related to the chromatin structure that will loosen up the chromatin, right, therefore we have to recruit acetyl groups. So what is an acetyl group? An acetyl group is basically this. So if we add this to a histone, what's going to happen? So what do histones do? Histones, so opposites attract, right? So histones attract the DNA to itself. So when we add acetyl groups, essentially what we're doing is that we're loosening up the DNA, right? Acetyl groups loosen up the DNA because they're negative, because opposites attract and like repels, right? So we're loosening up the DNA by adding an acetyl group. So let's read some of these answers. So the A is the correct answer. I'll just say that. Right. So let's start from the bottom. D is saying the activator will recruit histone deacetylase and the chromatin will be loosened. So loosening the chromatin is what is our final objective. That's what we're doing. However, just looking at this term, histone deacetylase. What does deacetylase sound like? It sounds like something that removes an acetyl group. So basically what that's going to do is that's going to repackage the DNA. It's going to tighten up the DNA around the histones. And then, so C, so this would be wrong, right? So now C is asking, the activator would recruit chromatin remodeler to wrap DNA around histones. Well, I don't think those exist. I don't think we went over chromatin remodelers, so don't worry about that one. And then the B is saying the activator will recruit histone acetyltransferase, and the chromatin will be condensed. Well, like we said, chromatin loosened is our final objective, so we don't care about our chromatin being condensed. So therefore, A says the activator would recruit a histone acetyltransferase, and the chromatin would be loosened. So histone acetyltransferase, meaning that an acetyl group is transferred to the histone, therefore loosening up the DNA, therefore enhancing transcription at that region. So that one's a really hard question to get a grasp of. Just my advice, if you didn't know what histone acetyltransferase is, or if you didn't know what histone deacetylase is, just use context clues. Is my advice. Just use your context clues because yeah, if I'm being honest, this test is not your friend. It's not gonna play nice. So yeah, let's go ahead and move on to question 16. Okay, so question 16 is one that I got a lot of questions about. So let's take care in how we answer it. So, Okay, I have a perfect chart for this. Not chart, but I have a perfect graph for this. So let me look it up just really quick. My cat has decided to make an appearance. So he likes to climb on my keyboard a lot, so yeah. Let's see. Okay, that's translation. Sorry, one second. Okay, I found the image. I probably could have found it quicker, but I apologize. Canvas is just so messy, and it's like I don't know. Anyway, so here pictured is the process of elongation during um, during transcription, right? So our RNA polymerase, our RNA polymerase is doing all the work. So it's unzipping the DNA, it's unzipping the DNA, it's adding RNA nucleotides, and it's proofreading itself as well. So basically, look at this. Look at which direction's being read. So we're moving this way, right? So then on the same scale, if we zoom in, right, we're still moving to the right, right? So 3 prime on this side, 5 prime on this side, right? And or it could be vice versa. You could have 3 prime on the other side, on the bottom, and 5 prime on the top. I don't know. It's just it depends on how or 3 5 on the bottom, 5 3 on the other on the top. Anyway, so look at what's going on here. So we have the 3 prime end and the 5 prime end. So we're moving this way, right? But look at which direction the mRNA is moving, right? So here we're going outwards, right? Or at least it looks like we're going outwards. However, what's actually happening is that we're making nucleotides this way, from 5 prime to 3 prime. So we're reading from 3 prime to 5 prime, and we're moving from 5 prime to 3 prime. Or sorry, we're making RNA nucleotides from 5 prime to 3 prime. I'm sorry, there's really just no way to no easier way to remember it. It's just how it is, unfortunately. So anyway, so like I said, we're reading in one direction, and we are making RNA. We're adding RNA nucleotides in the other direction. So what's one rule that we learned from sorry, what's one rule that we have learned so far? It's that DNA and RNA is always synthesized from 5 prime to 3 prime. Get this tattooed because you will need to know this always. DNA and RNA is always synthesized from 5 to 3, right? So, If we're always going to be synthesizing from 5 to 3, then we're going to be reading from 3 to 5 because we have to do the opposite. We have to move in the opposite directions, right? So we're reading one way and we're making the other way. And if we're always going to be making 5 to 3, then we're going to then transcription is going to occur along a 3 to 5 template forming mRNA in the 5 to 3 direction. So that's 16. So then moving on. So number 17 is asking, where in the RNA will you find a poly A tail? So for this one, you need to know certain transcriptional modifications or modifications we make to the mRNA. So mRNA modifications include RNA splicing, 5' cap, and 3' polyadenyl tail. So for RNA splicing, that includes introns and exons. So I'll also say this. All of this only occurs in eukaryotes. All these only occur in eukaryotes. Because transcription in prokaryotes happens at the same time as translation, so none of these modifications need to happen. Or none of them can happen, rather. In any case, let's go over some of these, right? So the one we care about for this question is the poly A tail. So basically, right, let's say we have an mRNA. This is our mRNA, right? So we have the 5' end and then the 3' end, right? On the 5' end, we have what's known as a 5' cap. So the 5' cap essentially acts as like a helmet. So if we have incoming like molecules that are trying to like they're like have a risk of like damaging the mRNA in sort of any sort of way. Well, if we're damaging from this way, right, if we're going to cut off nucleotides, well, we have our helmet to protect us from any sort of modification on that side. So that's what the 5' cap is for. And the 3' tail, the poly A tail, is basically what these are doing. As that these poly A's, these multiple adenyl groups, so it's the same A as in A, T, C, and G, right? So when we say poly A tail, these don't code for anything. So it's the same thing as the 5' cap, is that if one gets cleaved off, then it's not a big deal. It was meant to do that, right? It's meant for the protection of the mRNA, because if we cleave off here, well, that's really bad, because we cut off some of the mRNA, and therefore cut off some of the protein. So, but if we cut off here, well, no biggie, because these didn't code for anything, right? So these occur at the 5' end. So going back to number 17, the answer for this one would actually be A. It would be, you would find the mRNA, sorry, in the mRNA you would find the poly tail at the 3' end, because the 5' end we have the cap, introns are not related to this. Small subunit, once again, not related to this, that's on the ribosome. And then promoter, that's not really related to the actual mRNA. That's related to that's related to the region on the DNA. So what are introns and what are exons? So basically, and I know it's hard to because you'd think it's the opposite for this example, right? But exons, they stay in. introns go out. Right, so let's say we have the mRNA. So this is our unprocessed mRNA. This is just like a newly formed mRNA, just straight from the DNA. So, basically, exons are actual coding regions for the protein that we want. Introns are just stuff in between that we don't care about. So let's say the green regions are introns and the blue regions are exons. So basically what's going to happen is that we have an enzyme that's going to come in and it's going to remove these green regions. Right? So these are now going to be removed. So this is known as RNA splicing because we're literally splicing together the RNA using an enzyme. I think it's called a spliceosome, actually. And then we're going to make one continuous coding region, one continuous mRNA that has one long coding region. No nonsense in the middle. However, and this is why RNA splicing is so important, what if we only wanted the green portions? Well, we could do that too. So now the blue are the introns and the green are the exons. So therefore, all of these are going to be removed by a spliceosome, and the green regions are going to be glued together to make one new mRNA. So notice how for one RNA, we were able to make two different proteins. Right? So that's why RNA splicing is so important, so we don't have to have so much unnecessary DNA, because we could just cut around the DNA that we already have and make proteins that way. So that's what RNA splicing is. So introns, exons, and then for the sake of this question, it was the 3' end. So yeah, I think there's like a couple of questions about introns and exons. In any case. And I messed it up. Great. Alright, number 18. Sorry, I had to sneeze. Okay. So number 18 is asking, which of the following statements correctly describes the role of a molecule, e.g., the amino acid tryptophan, that controls a repressor of a repressible operon, like the trip operon? So, we already went over the trip operon, right? So we add tryptophan, and that stops transcription of that particular gene because we already made a lot of tryptophan, right? So tryptophan, it works. So like the operon is always on, but then tryptophan comes back to repress the protein. The tryptophan comes back to activate the repressor protein and shuts off the operon. So just based on that, the answer would be A, right? Which, because of course that's what's bolded here. So A is saying the molecule binds to the repressor protein and activates it. So here's some of the language disparity. The molecule binds to the repressor protein. So what's being activated? The repressor protein, not the operon. The operon is being repressed by the repressor protein, which has been activated by the molecule. Right? So that's what's going on in this case. A lot of people answered B, which is incorrect. Right? So B is saying the molecule binds to the operator region and blocks the attachment of the RNA polymerase to the promoter. So this is really tricky, right? Because the molecule is not binding to the operator region. That one's not true. However, the repressor protein would block the attachment of the RNA polymerase to the promoter. So, if we combined A and B, we can therefore say the molecule binds to the repressor protein and activates the repressor protein, which goes on to block the attachment of RNA polymerase to the promoter, which therefore shuts off the operon, inhibiting the production of tryptophan within the cell. So that's like, so that's just going off that. That's like the entire explanation of what's going on with the operon, right? So the molecule itself is not going to bind to the operator region. The repressor protein is. The molecule is going to bind to the repressor protein to activate it. In this case, anyway, right? Because we're activating the repressor. So in this sense, we can consider tryptophan to be what's known as a cofactor. Because we call it a cofactor because it needs to work together with something else. Hence, co. Co means with or together, right? So tryptophan acts as a cofactor in this sense, right? So C and D say the molecule binds to the promoter to decrease the affinity of RNA polymerase for the promoter. Well, that one's not right. Right, for one, it wouldn't bind to the promoter, like we said. Like, it wouldn't bind to the promoter; it binds to the repressor protein, right? And it doesn't decrease the affinity of a polymerase. It just would just straight up block it, right? And then D is saying, the molecule increases the production of inactive repressor proteins. Well, that doesn't really make sense. In this case, that doesn't make much sense, because why would we increase the production of inactive repressor proteins? Right? That doesn't really do anything. And we're not repressing anything by doing that. We're just increasing the number of repressors, which isn't really doing much. So, the answer is A in that one. So we are currently running at 1 hour and 17 minutes. We have about, I don't know, 10 questions left. Well, the last portion is the true or false, which hopefully shouldn't take that long. And these next two questions should hopefully go by quickly. So, number 19 is saying, what effect does methylation have on DNA? A, it allows it to be unwound in preparation for DNA synthesis. B, it makes it transcriptionally inactive. C, it activates transcription. Or D, it allows to be replicated nearly continuously. So we'll look at what these answers have in common. Allows it to be unwound in preparation for protein synthesis, activates transcription, replicating nearly continuously. All these are good things, right? We want these things to happen. So these are like enhancers, right? However, these are all wrong. Oh, yeah, they're all wrong. What methylation actually does is negative to the DNA. Methylation is pretty bad. So methylation is what's known as an epigenetic modification. So basically, what that means is that once we have our genome, and some certain parts of our genome have been methylated, those portions become transcriptionally inactive, right? So you can see why that's really bad. This happens throughout your life, through external factors, and unless they are germline, they are not transferable to your children. So typically these happen somatically. So essentially what happens is if we have our DNA, right? I don't want to make it too long actually. So if we have our DNA, and let's say on this end we have 3' to 5', right? So then we have the RNA polymerase binding to the whole thing, and then we're reading it this way. Well, if we have methylated DNA, essentially what that means is here on the outside, we have this sort of like flag. So once we get to this point, we can no longer pass. RNA polymerase can no longer pass this point because it's acting essentially like a roadblock. That's what DNA methylation does. Is it makes something transcriptionally inactive. We can't go past this point, so then our mRNA is not going to be good. It's going to be bad. Yeah, it's really scientific. So this would be an example of epigenetic modification. So this would be not necessarily a mutation because we're not actually affecting the DNA itself. However, we are affecting the transcription, which is bad still, right? So that would be considered epigenetic, which we will learn about more at the end of the semester. Number 20 is asking, what is the name of a type of protein that binds to an enhancer? So, what is an enhancer? So, an enhancer has to do with transcription. It has to do with like enhancing the transcription of a gene, right? So, what does that mean? So, if we're enhancing the transcription of a gene, right, we're gonna be so if we're enhancing it, then we're not going to be repressing it, obviously. And if we're and since we're not enhancing necessarily RNA polymerase, because RNA polymerase is doing its own thing, right? It's going to come in after the fact that we after so RNA polymerase. Okay, let me start over. An enhancer increases transcription. So a protein that binds to an enhancer would be an activator, hence activating the enhancer, right? Because sometimes we don't want to have extreme amounts of transcription going on. So it wouldn't be RNA polymerase, right? Because RNA polymerase actually makes the mRNA, right? So that wouldn't bind to an enhancer. It would be an activator that binds to an enhancer. So once this enhancer is activated, then RNA polymerase knows to go in overtime, start to kind of clock in and go overtime. That's essentially what's happening. And obviously, if we're enhancing something, we're not repressing it. That should be obvious. So that's number 20. I just realized there are there are two number 20s, and I'm sure many of you realize that too. So number 20. Let's call this number 20.5 or 20.1. That makes more sense. 20.2. That makes even more sense somehow. Wow. Anyway, so number 22, so number 20.2, sorry, is asking when different exons are combined to form different mRNAs from the same gene, this process is called, so remember what we learned from earlier, that is called splicing, right? When we're adding exons, that's called RNA splicing, right? So we're not worried about transcription, we're not worried about translation, we are splicing together exons to form different mRNAs. So this wouldn't be independent splicing because I have never heard of independent splicing, and then hopefully you haven't either because I don't think it's a thing. So the answer will be alternative splicing because we're making alternative RNAs from one gene, hence alternative splicing. So I had a lot of questions about this one too because this one is a little bit confusing. So number 21 is asking, in what ways are alleles of the same gene different? A, the two alleles have different DNA nucleotide sequences, or B, the two alleles control different physical traits, like hair versus eye color. So, remember, we get one set of chromosomes from dad, the other one from mom, right? So we have the red for dad, and then yellow for mom. So when we're talking, excuse me. So basically, sorry about that. When we're talking about alleles, we're talking about the same regions, the same coding regions on two homologous chromosomes. So we're talking. So remember, homologous chromosomes are pairs, right? So when we're talking about alleles, we're talking about this region, for example, and this region, for example. So these are the same genes, right? Because they technically they count as one pair, so they act in accordance with each other. I mean, you have 46 chromosomes, but really only 23 are working. Uh, don't take my word for that. That is just like a huge generalization. But basically, what is happening here is that we have two of the same coding regions, right? However, both of these coding regions will code for the same physical traits. So it's not like this one codes for eye color and then this codes for hair color, because that's not the case. Because down here we would code for hair color, or down here we would code for eye color. Over here we're coding for the same thing. So let's say we're coding for eye color here and here, right? So over here, dad has brown eyes, mom has blue eyes. So then dad wins because of dominance, which more has to do with genetics, but so we won't worry about that. So dad has brown eyes, so that we pass down as brown eyes, right? So that's alleles, right? The same coding regions. So notice, right? If dad has brown eyes on this allele and mom has blue eyes on this allele, well, that means that these two regions cannot be identical. I mean, they're the same coding regions. They code for the same physical trait. However, they're different. So therefore they have to have something different about them. So what is it different about them? The nucleotide sequence. So the answer would be A. So number 22 is quite long. At least the question itself is quite long. So, number 22 is saying, at the beginning of the century, there was a general announcement regarding the sequencing of the human genome and the genome of many other multicellular eukaryotes. There was surprise expressed by many that the number of protein coding sequences in the human genome was much smaller than it had been expected. Which of the following statements explain why there are more proteins than the number of protein coding genes found? So remember earlier, right? Remember in the case of that one mRNA, right? Right? So this mRNA can go through splicing. So we can splice out the green bits, leave only the blue, right? So that's one mRNA coding for one protein. Or we could splice out the green bits, we can splice out the blue bits and code for only the green, leaving another mRNA. So for one gene, that encodes for more than one protein. So the answer is going to be A, because this has to do with RNA splicing, alternative splicing, and how we get multiple proteins from just one gene. So B is saying some proteins are not encoded by DNA. That one is impossible. Do not focus on that one. That one is very wrong. This ignores the central dogma of biology. C is saying researchers have mistaken the number of proteins that have been made. That is also not true, because that's just not true. because a ton, literally in the Human Genome Project, billions, maybe even like $1 trillion went into the actual research of sequencing the genome. I don't know about $1 trillion, but it was a lot of money. Sorry, did I say $1 billion? It's just say $1 trillion. That's ridiculous. It was about $3 billion, which by scientific standards that is a lot of money and a lot of time. But by like I don't know actual funding standards, and I'm digressing. Sorry, researchers have not made mistakes in this question anyway. They have, of course. Once again, I'm digressing. I'm sorry. Okay, so in this question, this is not the right answer. And proteins have longer lifespans than DNA. That is not true. Typically, DNA is going to last longer than a protein because of how much better protected it is, right? So number 23, moving on. I had a lot of questions about this one too. So this one requires you. Sorry about that. This question requires you to know what a poly A tail is and how prokaryotic cells and how gene regulation differs. So anyway, so how is gene regulation different in prokaryotic cells versus eukaryotic cells? So in eukaryotic cells, we have the RNA, the journey of the RNA. In eukaryotes, right? RNA travels from the nucleus to the cytoplasm to the ribosome. So now, in prokaryotes, we go straight from the DNA to the ribosome. So notice this difference, right? So transcription and translation in prokaryotes happen at the same time, right? They happen at the same time. So there wouldn't need to be any sort of modification being done to the mRNA in a prokaryote versus in eukaryotes where we have to go from the nucleus outside in the cytoplasm where there's like a bunch of stuff floating around and a bunch of stuff that's about to like start degrading the mRNA, which then has to go into the ribosome. So some of these modifications, like we went over, include the poly A tail and the 5' cap. So do you realize why the prokaryotes don't need that? Because it doesn't need to be protected from anything, right? So there's no RNA that needs to be protected. So looking at some of these answers, right? Coordinated expression. So what I just described of transcription and translation happening at the same time, that is what's known as coordinated expression. So that would be seen in prokaryotic cells. Inhibition of transcription initiation, that one happens in prokaryotic cells, right? We went over operons. And the initiation of translational inhibition of translational initiation, that one also happens because transcription and translation happen simultaneously, so they have to happen together. So if we inhibit transcription, then we're also inhibiting translation, right? And then phosphorylation of a protein to make it active. Well, that happens in every single cell. That's just what phosphorylation does. Um, remember kinases? Yeah, that's what a phosphorylate. That phosphorylates something. That's what it does. Either make something active or deactivate something. In any case, what are we talking about? Poly A tail doesn't have to exist on an RNA and a prokaryote. because it simply doesn't need it. Hence, E is the right answer. So, number 24. Number 24 is saying, imagine that you have isolated a yeast mutant that contains a constantly active histone deacetylase. What phenotype do you predict from this mutant? Once again, I'm really sorry. I'm quite sick right now, so it's been a bit of a rough day. Um, in any case, we could continue on with the lesson. Um, so, do you remember that conversation we had from way earlier? Histone deacetylase, what an acetyl group does, and histone acetyltransferase. You know what that does? So acetyl groups, they will loosen the DNA, right? They will loosen the DNA because they repel the DNA. They repel the DNA from histones because negatives, because they're both negative, right? So like repels. So if we have a yeast mutant that is constantly deacetylating histones, if we're constantly making the DNA tighter and tighter with, so that's what deacetylase does because we're removing acetyl groups. If we're constantly removing these and we're constantly removing acetyl groups, right? What's going to happen? We're going to tighten up the DNA. And what happens when we tighten up? What happens when we tighten up the DNA? Well, then the RNA polymerase can't go in and start doing its thing, right? So the mutant will not grow rapidly. The mutant will not require glycose for growth because that doesn't really have anything to do with what we're talking about. And the mutant will not show high levels of expression. It will show very low levels of expression because we're tightening up the DNA. We're locking it down like Fort Knox with this deacetylase. So the mutant will show low levels of gene expression. Therefore, so number 25. So notice that there wasn't a lot of discussion about mutations in this press exam. My advice would be to go over those a little bit just to know like the difference between the frame shift and the point mutations, right? In any case, so remember, somatic mutations are not transferable to your children. Germlines are, because when you have a sperm cell or an egg cell that are mutated, well then your child's going to exhibit mutations, right? So so let's see. Number 25 is asking, occasionally a person who has one blue eye and one brown eye, also known as heterochromatia, who has a sector of one eye a different color from the rest. So, assuming the differences occurred due to mutation, in what type of cells must mutations have occurred in genes encoding for eye color? So right off the bat, a germline mutation will not affect this individual because it will affect the children of this individual. A somatic mutation will not be transferred down from the individual parents, right, which we just talked about, and then that leaves germline and somatic. It's germline in the parent and somatic in the individual. So look at what's going on here, right? One blue eye and one brown eye. Or has a sector of one eye different color from the rest. So this one is your primary hint, right? One sector of one eye different from the rest. So if we have one sector of one eye, so that means somewhere along the line the eye was normal, right? So that would have to happen after the individual was born, after the individual was conceived. So that would mean that a germline mutation is not on the table. So a somatic mutation would have to be the case for this example. So going a little bit over about mutations again. So we have a few different mutations, right? We have the point insertion, the point deletion, right? Those are known as frame shifts. And then the point, and then we have the and then we have like the single base pair ones where like they either they could be silent mutations or they could be missense or nonsense, right? So just a quick review. So we have point mutations, silent, missense, and nonsense. Now what these do, silent, no effect on the final protein. So that means it'll code for the same amino acid, kind of like how we went over way earlier. Missense means that that one amino acid is coded incorrectly. So if we have like AUG, if we accidentally change a U to a G, so AGG, then that one amino acid is changed, so that would be a missense mutation. So when we're talking about point mutations, that's one amino acid that's being affected. Or one, sorry, when we're talking about point mutations, that is one codon that's being affected. So let's write that down. Now we're talking about nonsense, right? Codon now codes for a stop codon. So do you see how some of these would be, how we're going from levels of severity downward, right? Silent wouldn't be severe at all. That would be have no effect, right? Missense would be kind of bad depending on what the amino acid is, right? If we're changing one amino acid in one giant protein, well, there's still a little bit of room for error. However, if the codon now codes for a stop codon, if we're ending earlier than what we started with, well, that will be really bad. And that's why I think that's the reason why sickle cell anemia operates the way it does. It's because the codon, because the production of a hemoglobin stops early, so it ends up with a sickle cell looking. Kind of like that versus the normal blood cell, right? So that would be nonsense, right? That one's really bad. We don't want that. So then we have frame shift mutation. Oh, sorry, then we have frame shifts. So now frame shifts are really, really bad, just like nonsense mutations. Frame shift, meaning that more than one codon is affected. Here we have point insertion and point deletion. So point insertion is when we add a nucleotide where it doesn't belong. And then in point deletion, we delete a nucleotide from the DNA. So, in doing either one of these, we are shifting the entire, like if we're making an mRNA, right? We are shifting all of the codons by one, right? So if we have like AUG, TAC, AUG, UAG, GGC, and if we add a U at the beginning, then it becomes UAU, and then sorry, I don't remember. It was like GUA, I think. I don't know. Anyway, it shifts one more over, so that you know how that. So you see how that would be really bad because everything is messed up, right? And the same case for deletion. AUG is now so AUGCCA is now UGC whatever. So the ones that affect multiple codons, those are the ones that are really, really bad. So that's just a little bit about the mutations, right? Silent, that's like the best case scenario. And missense, that might actually be beneficial. So that one's like iffy. But in most cases, nonsense and frame shift mutations are the ones that are going to be really bad. Anyway, let's do a bit of a lightning round for the last five. So, number 26, ribosomes on the side of translation. That is true. We should hopefully know that by now. That is true. Number 27, the genetic code is redundant. More than one amino acid can be specified by one codon. That is false. Do you know why that's false? We'll read it again. More than one amino acid can be coded by one codon. It's actually the opposite. More than one codon can be specified by one amino acid, right? So if we have a codon, well, codons like, for example, okay, let's pull up the codon chart, right? Well, if we have a codon chart, right? Look at what we have: GUA, valine; GUG, valine; GCU, alanine; GCC, alanine. You see what I'm getting at here? So we would have more than one codon. So the genetic code is redundant, right? We understand that. However, it's not because more than one amino acid codes a specific codon. It's that more than one codon specifies one amino acid. So this answer would actually be false. So number 28, euchromatin is loosely packed chromatin readily available for transcription. That is true. Now, what is euchromatin? Eu means true or pure or whatever. So euchromatin, so that would have to mean that the chromatin is already available for transcription. That's essentially what that means. Number 29, tRNAs transfer amino acids to the growing polypeptide in a ribosome. That is also true. That happens at the P site. Remember EPA site, right? Well, when we have the ribosome, so remember the ribosome has three different sites: E site, the P site, and then the A site. So then we have the tRNAs. So let's draw. Oh, hold on. This is our mRNA, right? So then we have the tRNAs, and with an amino acid, they go at the A site. So A for acceptor, P for processing, E for ejection. They go to the acceptor site. They get processed at the P site where they elongate this amino acid chain, and they get ejected at the E site where this tRNA then goes on to get regenerated. Hence adding an amino acid to the amino acid chain that is currently growing. So tRNAs transfer amino acids to growing polypeptides in a ribosome. That is true, right? So then, last one, spontaneous mutation can occur due to DNA replication errors. That is also true. So most mutations are already spontaneous, right? I mean, you have a higher chance of getting mutations via like mutagens or carcinogens. However, spontaneous mutations typically do occur due to DNA replication errors. So, like I said, so DNA replication errors happen once every 9 billion base pair additions. Sorry, that's not right. 1 in 1 nucleotides that are added are errors. So do you see how they could be spontaneous? It just happens randomly. But it's really, really rare. But just think about how many nucleotides you have in your cells, right? A lot. So 1 in 1 billion doesn't sound like too infrequent now, does it? In any case, that is your practice exam. So, my final advice would be to 1, study up your slides. 2, study up your slides, and 3, study up your slides. What I'm trying to get at is that your slides are where the content is going to be drawn from the most, right? Your slides, that's what counts for the most, right? I mean, the study guides don't really help you, do they? I mean, the study guides are like they're taken from Dr. Howe, so they're not written by Dvorka. So they're going to be tailored to Dr. Howe's exams, in which you're not taking Dr. Howe's exams, are you? So my advice: look at the slides because anything is fair game on the slides, no matter how difficult it may be. But hopefully, I helped you out a little bit with this little review. We were here for quite a while. It's been about an hour and 50 minutes. Um, so if you're having trouble on one particular question, I hope I answered that question. In any case, please feel free to email me if you have any questions, and I wish you all the best of luck on your exam tomorrow. So I bid you all farewell. Have a good one.